Hello and welcome to the VMOLS tutorial. This will teach you how to use the VMOLS program. VMOLS stands for Virtual Molecular Modeling Kit. This was created by Professor Rathenberger and Mr. James Webb, formerly from the Illinois State University, and they've made this program available online for free to whoever wants to use it. So you're going to open up your lab. You're going to scroll through to the part where it says website link 3. This is the actual program. I'm going to hold my control button down and click it, and you'll see the program opens up. And in this case, since the last molecule I analyzed was this particular molecule, this is vitamin B1 thymine. This is what you'll see. In your case, you're going to see aspirin, and so I'm going to select aspirin here. But if you want to put any new molecule in, you're going to click the name button, and this is what you're really going to do first in any case, name button, and you've got lists of various molecules you can choose from. You're going to pick 3 molecules, and let's just try typing in a new molecule. So I'll just type in Tylenol, for instance, and here's the structure for Tylenol, also known as acetaminophen. And I can take my mouse and I can rotate this in space, and I can rotate it left or right. I can rotate it vertically. I might even be able to rotate it slightly diagonally, but you can get it into an orientation that looks good to you, so you can see all the atoms and all the bonds. And this particular model is called the ball and stick model. It's listed as ball here. You'll see 3 different models listed: wire, ball, and space. Wire model looks just like straight wires. You don't see balls of atoms, you just see the bonds and the color coding for the atoms. So let's go back to the ball. We're not going to use the wire too much. I don't recommend it. And in this system, gray represents carbon, white represents hydrogen, oxygens are red, nitrogens are blue. And this is pretty standard in most molecular model kits. They might vary. The carbons, they might be black in some kits, but in this kit the carbons are gray, but you'll almost always find the oxygens are red and the nitrogens are blue. And you'll find other atoms or other colors. If you ever get confused as to what the other atoms are, the ones that aren't nitrogen or that aren't hydrogen or carbon, what we call the heteroatoms, you can just click this stereo on-off button, which brings it into a 3D mode. If you were wearing 3D glasses, you might be able to see this in 3 dimensions, but what we're gonna use this button for is it labels the heteroatoms, the oxygens, the nitrogen, carbon, not carbon and hydrogen, but it will label the halogens, fluorine, chlorine, bromine, iodine. It'll label boron and silicon, phosphorus and sulfur if you get confused by the color coding. And we're just going to toggle that off to get back in the view. So this is the ball and stick because the atoms look like balls and the bonds look like sticks, and you can see the bonds are either single bonds or double bonds in this case. In some molecules, you'll see triple bonds. But if you go to what's called the space-filling model, which is right here, this is based off of space-filling models that were developed by 3 very famous scientists, and they're abbreviated for their names. They're called the CPK models, and these are closer to what the model of the molecule actually looks like. It's not exactly, but it's closer. And you'll notice you don't see the individual bonds in the space-filling model, but it does show you essentially how fat the atoms are as they're pressed up against one another, but you can't tell whether they're double or single bonds or even triple bonds unless you go to the ball model, and then you can see the actual bonds again. Now, we have a number of different functions. If you want to blow the picture up, you can do model zoom plus, but then you get too big, and you can go model zoom minus. We can rotate it as we already showed. And if we click the move button here, there are three different options within the move button. There's the translate, which just means move around in the box. If I double click inside, it says rotate, and you've already seen the rotate mode. And then the third mode is drag mode. And please do not use the drag mode, because if you do, you'll end up distorting your molecule and you have to start all over again. But if I click again and I hit translate, and if you put your mouse right on an atom, instead of rotating it, I can actually move the whole molecule to a different part of the box if I want to. And so I can still grab outside and rotate it, or I can grab on the atom and move it in the translate mode. So let's get out of that. So we can undo most of these things by hitting clear reset. It will not undo the drag function. If you distort your molecule, you'll have to retype in your name and start over again. But we have our molecule in here. Maybe we want to show it in a different orientation. I can rotate it around. It takes a little practice to rotate this into positions you like. So just play around with it. And you will notice that in addition to being able to move it and rotate the molecules and blow it up and shrink it, we can also analyze the molecule. And the most useful functions are down here where it says other model actions. Charge, dipoles, dipole net. We're going to skip energy. VDW, which stands for van der Waals view, or van der Waals radius view. And MEP, which stands for molecular electrostatic potential, and I'll explain what these are. So if you click on charge, it will show you the partial charges of all the atoms. Remember, this is a molecular compound, a covalent compound, so there are no full-blown charges, but there are partial charges, as we learn when we study electronegativity and dipoles. So this shows the partial charge. So you'll notice this oxygen here has a partial charge of negative 0.57, not a full -1 charge. And if we click that again, we can momentarily make it go back, but if you really want to make it go back, click the reset button. That'll be your best option here. So we can look at charges. We can look at dipoles. So dipoles are how pairs of bonded atoms are related to one another in terms of electronegativity. So if one atom is more electronegative than another, in this case we have a carbon and an oxygen, and the oxygen's more electronegative, we'll get a dipole going from the carbon to the oxygen, and that's represented by this red arrow. And the bigger the arrow, the bigger the dipole. We have another dipole here between the carbon and the nitrogen in blue. So oxygens are going to be in red, nitrogens are going to be in blue. You can always go to the stereo view, and that will tell you, again, which atoms it is. And so we can go back to the dipoles and look at individual dipoles. If you don't see arrows, or the arrows are very short, that means they're very small dipoles. So most of the dipoles are around these colored atoms. If you want to look at the overall dipole for the whole molecule, so if you add up all the individual arrows, you get one arrow that represents the dipole for the whole molecule. And this arrow is moderately long. It's not enormously long. So this tells you there's a moderate dipole, and it gives you some idea of the polarity of the molecule. I'm going to undo this by hitting the clear reset. But if you really want to know how polar the molecule is, the better way is to use what we call the MEP mode, the molecular electrostatic potential. And this is just a fancy way of saying, and you got to wait, where are the partial charges? And this tells you, it's kind of a map of the surface of the molecule, and it gives you kind of a three-dimensional shape of the molecule. And red areas represent partially negative charges, and you can see down here it says partially negative for the red, and the blues represent partially positive, and the greens are somewhat neutral. They're non-polar in the middle. So you can see this molecule has a lot of red and a lot of blue in it, and that tells you that the molecule overall is fairly polar. And I'm going to undo this. If you just want to look at the overall shape of the actual molecule, you hit the VDW, the van der Waals form. And this van der Waals view really shows you the shape of the molecule as it appears to neighboring molecules that might bump up against it. So this is the actual shape. It's not nearly as pretty as you might imagine. It's kind of blobby in shape, and that's because the atoms aren't points in space, and the electrons are moving around, and they occupy these volumes of blobby space. So the van der Waals the van der Waals surface really gives you an idea of the real shape of the molecule. So we're going to get out of that again. So we have these different options for viewing it. And these are very useful for analyzing. If we want to look at the length of a particular bond, we click the length button, and then we click two atoms that are bonded to one another. I'll click that one, and I'll click that one. And you can see it tells me the length is 0.123 nanometers. Nanometer meaning billionth of a meter long. So these are very small molecules. The molecules are very tiny for the most part. If I want to look at another, I can click and get the distance between the nitrogen and this carbon, 0.14 nanometers. So I can clear that. If I'm interested in the angle between three atoms, I click angle, and I'll click 1, 2, 3 bonds, and it will tell me the angle between these is I don't know why it's blinking on and off. Let's try this again. I'll click, click, and click. Sometimes it doesn't work. We'll reset, and we'll do it again. Click, click, and click, and there it is. 127.4 degrees. So that's about a third of a circle, which is what you'd expect in this case. So we're going to clear that. And we also have other functions, mass, if you want to look at the total mass in grams per mole for it, but you don't need to do that for this particular lab. But we also have this really unique function called rotate bond. If I click this, and then I click a bond, so I'm going to click that bond, and you'll notice it highlights the atoms that surround that bond. And now if I take my mouse and I rotate something not directly on the bond, I can actually rotate just part of the molecule without rotating the entire molecule. I'm rotating around the bond that I selected. And this really only works on most single bonds. It won't work on double bonds because double bonds can't usually rotate. The same with triple bonds, not easily. So this is really useful for this rotation around a single bond. If I wanted to change what bond I wanted to rotate, let's see what happens on this one. It's within a ring, and it highlights it, but I don't know if I can rotate it much. I'll try rotating it, and you'll notice it doesn't really allow me to twist the molecule. It just allows me to rotate the whole molecule around that bond. This single bond is within a ring, so it's not going to twist too much. So we'll undo that. So you have all these different functions for analyzing your structure in terms of lengths and angles and charges and dipoles and overall shape and polarity. And this is what you're going to use to analyze your molecule. And when you're done, let's say you're happy with this and you want to put a picture of this in your lab, you will click the button Save PNG, and this is an image file, and you don't have to name it. You can just use this default naming, but it'll overwrite all your pictures. So I can just name it whatever I want, but I'm going to leave it alone. I'm going to hit OK, and that just names it, and now it saves it to my computer, and I'm going to open up the file, and this is how you will actually copy and paste your picture. The file will open. You'll notice it's just an image of the last manipulation you did of your molecule. I'm going to right-click my mouse, select copy, and now I'll get out of the program, and I'll go back to my lab, and my lab where I'm working, I'm going to click on the page in the lab, and I'm going to paste my picture onto the page. And usually it's going to be too big, so I'm just going to resize it and move it in place, and then I'll just save it, and that will be in my lab. And now I have it there, and I'll name my molecule, whatever that was, and I'll write the findings that I found. I'll talk about the functional groups. In this case, this molecule has a an amide function here, an alcohol function here, and an aromatic ring. So I'll talk about that. I'll talk about the fact that the molecule is fairly polar and that it has a moderate dipole for the whole molecule and several dipoles around these different, the carbon double bond oxygen and the carbon single bond nitrogen, and this carbon to the oxygen and this oxygen to the hydrogen. They all had dipoles in it. And I can talk about how the van der Waals surface showed me that this hole is not as big as it looks like in the ball and stick model. So there are lots of things you can talk about in here in the interesting findings. And with that, you're ready to start your own analysis.